everybody, welcome to a video of Owl Steam. So in this video, the first thing you might notice is that I have a huge amount of items on my table. And today, our experiment is something that I've been waiting to do, and that is to extract DNA from blueberries. So these are all the ingredients you'll need. You'll need some blueberries, of course. You'll need a chopstick or something to get the DNA, a measuring cup of water, a strainer, a spoon, an isopropyl alcohol which has been chilled, some dish soap, and a plastic bag. Let's get started. So first you want to open this plastic bag and put the blueberries in. Next you want to pour some of the water in. And wait a second, I think I forgot the salt. Okay, let me go get the salt. Okay, so now I'm back with the salt, and you can see I got the salt, and I'm going to reopen this plastic bag. I'm so sorry, I forgot the salt. Yeah, you need salt. Just going to open up this bag. I'm going to just pour some salt in. There. Then, this is going to be a bit tricky because I only have only myself, but I'm just going to somehow open this thing. Surface is bad. And I'm going to just pour some of this into there. Should be enough. And close it. And then we close the bag and make sure that there's as less air as possible. And when you've made sure that, that's a lot of air, but whatever. So now I have your plastic bag. Now all I have to do now is smush the blueberries. Now blueberries are actually surprisingly hard to smush, so because they keep sliding around, but just try to do it as best as possible. I think I may have given too much water, I don't know. But anyways, start smushing it and smush it until there's no large pieces anymore. Now blueberries have the skin on them, so if there's still skin, it's fine. But try to get as much of the meat or the other stuff as into this watery liquid thingy. And why is this thing open? Did I not close it correctly? I don't think I did. That would have been a disaster. Anyways, so once you feel like that you've smooshed this thing enough, I'm sure to shake it a bit. There's a lot of bubbles, so now. I don't think there was supposed to be any bubbles. And I think that should be enough. Now I take our strainer thingy, and I didn't notice that it fit exactly perfectly with the water thingy. Anyways, we open up our plastic bag, and we pour the contents on top of the strainer. And there. And, well, you don't need this plastic bag anymore, so I'm just going to set it aside. And we use the spoon and smoosh out as much stuff into this. So, I'm going to zoom in so you can see it better. So I'm just smooshing this strainer so that it all as much juice goes into the into this cup at the bottom, and that should be enough. So I actually don't know where to put this. I guess I'll put it here. I guess. Okay. And it's time for this. I'm gonna get my spoon again, and this thing's in the way. I'm gonna put it on the ground. And why is the table moving? Okay, anyways. I'm usually clumsy, but I'm just going to start pouring in some of this alcohol. And actually, I don't think I need this spoon. I'll just do this. Very carefully pouring in some alcohol. And oops, I spilled some. But whatever. And after some time, we'll wait. First, we'll let this thing sit for a bit. And I'll tell you when this is ready. So after a bit of waiting, this is what we've got. So, I'm not sure if you can see it, so I'm gonna zoom in. Do you see that like milky white, almost wow? That is no longer clear. What has happened? If you can see, like there's kind of like a milky white layer. See here, just below the bubbles, there's a milky white layer there. And that is DNA. Now you might be wondering, hey Albert, what is DNA? And that's what we're going to be talking about. But first, the link to this experiment with strawberries is included in the comments below. So be sure to check that out and see if this actually works for you. Okay, it's time for talking about what DNA actually is. Okay, here we go. So, we have DNA here, and yeah, you can see this is not a very accurate representation of DNA, but it's the best I can do. And then you might have noticed I've drawn AGTC here. You might be wondering, what is this AGTC for? Well, A stands for adenine, G stands for guanine, and T stands for thymine, and C stands for cytosine. I'm going to type it all out so you can understand. So adenine, guanine, thymine, and cytosine. Well, so what are adenine, guanine, thymine, and cytosine? Well, say if we were to like zoom in on this, and these thingies, these lines that are drawn, they're actually the bases. And base by and I mean base by one of these, AGTC, or adenine, guanine, thymine, and cytosine. Now you don't have to remember adenine, guanine, thymine, and cytosine, but it's really good to memorize them. But AGTC is the one that you really need to memorize. Anyways, back to this one. So each of these, each of these things is A, G, T, or C. So let's say that this one is A. And I don't think you can see that, so I'm going to turn down the thickness. That's A. Now, automatically, we know that this one is T. Why? Because A always pairs with T, and C always pairs with G. So if this one is G here, then this one is automatically C. It's kind of like quantum entanglement, where when you know one of them, you automatically know the other. But quantum entanglement is something that I'll talk about later, and you don't have to worry about it. So then say this one's A, T, and this one must be A. And this just goes on and on and on throughout the DNA strand. And, oh yeah, I forgot to talk about what DNA stands for. DNA stands for deoxyribonucleic acid. It's a very long word. So, deoxyribonucleic acid. So, that's DNA. Okay, anyways, So, next, let's talk about purines and pyrimidines. Now, purines and pyrimidines are things that I've gotten confused a lot, actually. Because, it's just really confusing which one is which. So, purines, and pyrimidines. So, let's take a look here. So purines are A and G. So adenine and guanine are the purines, and the pyrimidines are thymine and cytosine. Well, you might be wondering, why do we classify these two? Why do we classify A and G, and T and C differently? Well, the reason is purines and pyrimidines have a different structure. Purines have a different structure than pyrimidines, and vice versa. If you actually zoom in on adenine, what you see is there's this ring, one ring, and then a second ring. I think this one is the 6-member chain, or is it a 5-member chain? Let me check real quick. Okay, this one is not correct. It's actually a 5-membered ring and a 6-membered ring. So, like this. So that's what a purine is basically like. I'm not saying that it actually looks like this in real life. Of course it doesn't. But let's just do this. And then, so those are purines. Pyrimidines, however, only have one ring, and it's 5-ringed? Yeah, it's 5-ringed. I mean, it's 6-ringed. No, what? Is it 6-ringed or 5-ringed? Sorry. It's 6-ringed. It's a 6-ring, and it basically looks like this. So that is basically pyrimidines, which are C and T, and this is a purine, which is A and G. So enough about purine and pyrimidines, or whatever that is. And now we're going to move to the side. Also, one thing to note is that DNA is a double helix, meaning that there's two, like, spinny, spirally things. And, yeah. So now, let's talk about RNA. Now, RNA is different from DNA, and it's called ribonucleic acid. So it doesn't have the DNA, the deoxy part that DNA has. Now, this is basically what RNA looks like. Now, it doesn't exactly look like this, of course, but I'm just drawing it so that you get an idea. Now, if we zoom in, you will notice that we have A, that's the same, U, which is a bit weird, and A, G, U, and C. Well, you might not know what U is. U stands for uracil, so uracil. And I purposefully wrote it in the same exact place as I wrote T here in AGTC, and that's because uracil is nearly the same as, in fact, the only thing is that they're both pyrimidines, and the actual only difference between uracil and thymine is that uracil is missing a methyl group, which is which is a carbon and 3 hydrogens. And yeah, but we won't get into that. So uracil is so similar to thymine that people once thought, why not use thymine? Why use uracil? The answer to that question is because RNA existed before DNA. So technically, uracil actually came before thymine. And one theory suggests that uracil was easier to use than thymine, it was easier to use. For some reason, it was easier to use. All right. So now that we've covered RNA and DNA, we need to cover mRNA. So mRNA is a special type of RNA that looks exactly like this, and it's basically a messenger. So its name suggests that its name is messenger RNA, so obviously it's a messenger. It's produced by DNA using a thing called RNA polymerase, which I'm not going to go into depth about, but you can search it on your own. But I'll type DNA polymerase in the description below so you know how to spell it. So anyways, and then there's going to be a complicated chain of events that happens, which turns the messenger RNA into a protein. And that's what we're going to be talking in the next video. It's time for the question of the video! Yay! Okay. So, let's say that you have your DNA strand here. And let's say that your DNA strand has a mutation here, so that one of the bases is missing. So let's say like this. And then this base is missing. There's no base here. What is going to happen? Is the body going to replenish that base? Is it going to add that base in somehow? Like get a base from the another piece of DNA and put it in? Or what? What else is going to happen? Or is it just going to stay like this for the rest of its life? Or, well, it's not living, but whatever. Leave your answers in the comments below. Please like and subscribe. And I'll see you in the next video.